Bulk transport. All right, so let's talk about bulk transport. Bulk transport is a way to move lots of small molecules and water into the cell, or maybe out of the cell, right? And also large molecules like polysaccharides and proteins. So there's two major types of bulk transport. There is exocytosis and endocytosis. Both of them require energy. So these are forms of active transport. Exocytosis, you can kind of think of it like exiting the cell. So things are being exported out of the cell. Endocytosis is into the cell. Now let's exocytosis. Well, all of them kind of involve vesicles, but exocytosis involves transport vesicles that are going to migrate to the membrane and fuse with it and then release the contents from the cell. There are certain cells that actually do this regularly. They are secretory cells. Endocytosis is basically just the opposite. So the cell takes in macromolecules and other things by forming vesicles at the plasma membrane. So endocytosis is just basically the reverse of exocytosis, but involves different proteins. There are three types of endocytosis. There's phagocytosis, penocytosis, and receptor-mediated endocytosis. Phagocytosis can be nicknamed like cellular eating, and you'll see why in just a moment. Penocytosis is like cellular drinking, and receptor-mediated endocytosis is just something altogether special. So let's look at phagocytosis. Phagocytosis is when a cell engulfs a very large food particle. So it's, remember, cellular eating. What actually happens after that is it's like a vesicle, but it's larger, so it's called a vacuole. Okay, so a food vacuole forms. This food vacuole will eventually fuse with a lysosome, and then remember the contents of the lysosome, right? So that contains digestive enzymes and a very acidic pH, so it helps break things down. So there, we're eating and we're breaking the food down. Penocytosis allows the cell to create vesicles along the surface of the membrane to take in small molecules and water. So it's like drinking, right? So what forms with penocytosis is they're like coated vesicles. They've got proteins that coat the vesicles, and they're handled in a different way. Receptor-mediated endocytosis is very similar to penocytosis, except a receptor is required. So here we've got this little red receptor that looks like it's shaped like a Y. So it has a special shape. So the only thing that can bind to it is whatever fits in that little V part right there. So these little purple triangles fit in that. So those can be called what's called the ligand, Ligand. Ligands are just molecules that bind to receptors. So once this binds to the receptor, it induces the formation of a coated vesicle. They're usually coated with a protein called clathrin. Clathrin-coated vesicles, again, are handled differently than regular vesicles. So again, this allows the take-in of specific molecules. So if we think of phagocytosis and penocytosis versus receptor-mediated endocytosis, the difference is that receptor-mediated endocytosis is very specific. The others are more general. They just take in whatever, right? This is, it's taking in a specific molecule that binds to that specific receptor. All right, so this kind of summarizes bulk transport. So we have phagocytosis, cellular eating, a food vacuole forms, which is going to eventually fuse with a lysosome to break down that food. Here we have coated pits formed with penocytosis, except this is more like cellular drinking. So water and smaller ions and molecules are going to enter the vesicles and be taken in by the cell. And then receptor-mediated endocytosis requires a specific interaction between a receptor and a ligand, or a molecule that binds to it. Once it binds, it forms these coated pits, which will be handled in a different way in terms of metabolism. This summarizes transport. So remember, there's active and passive transport. Passive transport involves just simple diffusion. Part of that would be osmosis and facilitated diffusion, and I talked about tonicity. And then active transport, we've got the sodium-potassium pump, the proton pump, and then co-transport, and of course bulk transport, which involves exocytosis, or exiting the cell, or endocytosis, or taking in things by eating, drinking, or something specific. All right, thank you for listening.